everyone, my name is Usman Muhammad Shika, an MSc student 700 level Public Health of the JPTS Institute of Management, Science and Technology in collaboration with South America University USA and Global Wealth University Togo. I will be presenting a brief seminar paper on the theme the presence of hepatitis B envelope antibody in patients who have been previously screened for the surface antigen. The hepatitis B virus, HBV, is an enveloped DNA virus with an icosahedral capsid replicating via reverse transcription. The crystal of the capsid is known. It has a diameter of 36 nanometer and is formed by one protein species, which is C protein. The viral envelope contains three different coterminal proteins, S, M, and L proteins, spanning the membrane several times. These proteins are not only released from infected cells as components of the viral envelope, but in 10,000-fold excess as subviral lipoprotein particles with a diameter of 22 nanometer containing no capsid. Diamonds containing no capsid assembly of the capsid occurs in the cytosol and results in the packaging of a 3.5 kb ribonucleic acid molecule together with viral and cellular factors. So this newly formed capsid cannot be enveloped. Rather, synthesis of the viral DNA genome in the lumen of the capsid by a way of reverse transcription is required to induce the budding component states. Envelopment then takes place at an intracellular membrane of the free Golgi compartments, the S and the L protein, but not the M proteins. So this is required for this process. The L proteins forms two different transmembrane topologies. The isoform exposing the N-terminal part at the cytosolic side of the membrane is essential for budding. In this domain, a 22 amino acid AA long linear stretch has been mapped genetically to play vital role in the morphogenetic process. This domain probably mediates the contact to the capsid. A second matrix domain was mapped to the cytosolic loop of the S protein. A similar genetic approach identified two small areas on the capsid surface which might interact with the envelope proteins during the envelopment. Hepatitis B surface antigen, HBsAg, a protein on the surface of hepatitis B virus. It can be detected in a high levels in serum during acute or chronic hepatitis B virus infection. The presence of HBsAg indicates that the person is infectious. So there is yet no treatment for acute HBV infection, but it is a vaccine preventable infection because the real treatment is not up to now been achieved, but through some preventable vaccine it can be prevented. So there are several vaccines which have been used in the prevention of this infection, and the most prominent ones are lamivudine, adefovir, and diafaroxil, etc. So Thank you very much.